you about Christian Santamaria, who will be speaking with us, is that he actually started with the P30, well, not necessarily started, but worked with the P30 as someone who helped provide services. And so I've had the pleasure of knowing and working with Christian for a really long time in that capacity, and then watching him go through his DDS PhD. So Christian, I'm really looking forward to your talk. Wenhan.
Sure. So good afternoon, everyone. Welcome to the Hard Talks and Course Lines seminar series. And for who don't know me, I'm Wenhan Chang, a professor in the Endocrine Research Unit in the UCSF Department of Medicine, and a research career scientist at the San Francisco VA Medical Center. And I'm excited to introduce the first of a two exciting talk today. And
okay.
Sorry, we have a little bit of background. Can we somebody turn off the mic? Are we good?
Oh, people will mute.
Yeah, please. So sorry, it's my great pleasure to introduce our first speaker, Christian Santamaria. Christian is a practicing clinician scientist. He completed both his DDS and the PhD training at the UCSF School of Dentistry and the Oral and Craniofacial Sciences program 2 years ago. And his graduate and postgraduate work in my lab has been focused on the development of new regimen to enhance the current FDA-approved PTH-based therapies for treatments of osteoporosis and fracture repair. And he has received several awards for his translational research, including NIH F30 Fellowship Award, multiple Young Investigator Research and Travel Award from ASBMR, Endocrine Society, and European Calcified Tissue Society. In today's talk, he will present one of his studies demonstrating the synergistic osteoanabolism by co-targeting the PTH1 and extracellular calcium sensing receptor, and lay out a potential pathway to a new bone therapy. So Christian, can you go ahead, just take over?
Sure. Thanks for the invite to speak today and sharing your lunch hour with us here. All right. And so the mainstay of current therapies for osteoporosis are anti-resorptives, specifically the bisphosphonates, which work to slow bone resorption. Parathyroid hormone, or PTH1 at 34, and most recently abaloparatide, have shown to increase bone turnover, creating an anabolic window for new bone formation, thus increasing spine and hip BMD and reducing vertebral and non-vertebral fracture risk. However, there's certain challenges to intermittent PTH therapy. High doses, which can produce stronger skeletal anabolism, it can cause hypercalcemia, and side effects include headaches, muscle cramping, nausea, among other symptoms. So this is due to the supraphysiological actions of the peptide on the systemic calcium homeostasis. So by increasing calcium absorption in the gut via renal production of 125 vitamin D, increasing calcium reabsorption in the kidney, and mobilizing calcium from bone in the bloodstream. And that's how you get the hypercalcemic effect from PTH. And while it's not been shown in clinical studies, long-term use of high doses of PTH posed an oncogenic potential in rats. And for these reasons, the FDA only approved a conservative daily dose of 20 micrograms for no more than 2 years of treatment. This produces a significant but relatively low bone gain in trabecular sites and less apparent in cortical sites. Also, this anabolic effect can be difficult to maintain after treatment ends. So patients are then switched to an anti-resorptive, for example, to kind of preserve that gain in bone. And lastly, intermittent PTH therapy can be quite expensive. So our studies aim to optimize the PTH therapies by sort of mitigating the hypercalcemic side effect to allow for treatment with higher doses of PTH, and or the use of stronger PTH analogs, such as the reported long-acting PTH, to shorten the treatment period. So our strategy is is based on the interactions between the calcium sensing receptor and the PTH receptor in the context of calcium homeostasis. So we and others have shown that activation of the calcium sensing receptor counters the hypercalcemic actions of the PTH1 receptor by suppressing the endogenous PTH production in parathyroid cells, inhibiting calcium absorption in the kidney, and calcium mobilization from bone. Together, you kind of defend against hypercalcemia. And indeed, small allosteric agonists or calcimimetics are being used to clinically treat hypercalcemia in hyperparathyroid patients and with kidney diseases. So we and others have also shown here that the expression of the calcium sensing receptor are in chondrocytes and osteoblasts. They promote cell survival, terminal differentiation, and their mineralizing functions. So in vitro and in vivo studies have shown that this receptor is essential for perinatal bone development, as well as skeletal maintenance and repair in adulthood. The receptor is also expressed in osteoclasts and osteocytes, where its activation suppresses resorptive and osteolytic activities of the cells respectively. And so based on the abilities of activating calcium sensing receptors to defend against hypercalcemia and produce skeletal anabolism, we hypothesize that concurrent activation of the calcium sensing receptor with a calcimimetic drug can not only mitigate that hypercalcemic actions of PTH, but also synergize the skeletal anabolism to a level stronger than PTH alone. So to test this hypothesis, we injected 3-month-old male and 12-month-old female mice once daily for 4 weeks with vehicle, PTH, or calcimimetic alone, or in combination of calcimimetic and PTH. So we performed serum analysis for calcium and bone turnover markers, and also performed high-res micro-CT analysis and histomorphometry to observe skeletal changes. So to test this, so what we first tested was the acute effect of the treatment on serum calcium 3 hours after injection, specifically whether the calcimimetic can indeed offset that hypercalcemic effect at increasing PTH concentrations. So as expected here, you see that when you inject PTH 3 hours after, it produces a significant rise in serum calcium, while the calcimimetic lowers it, likely by suppressing the secretion of endogenous PTH from parathyroid glands and promoting calcium excretion in the kidney. Serum calcium in mice co-injected with calcimimetic at 10 and 20 micrograms per kilogram of PTH trend toward normal, while the hypercalcemic effect couldn't be normalized at the 80 microgram per kilogram dose. However, co-injection with calcimimetic at the 40 microgram per kilogram of PTH normalized serum calcium most effectively. This combination was essentially like our sweet spot here. We use this for subsequent skeletal assays. And these studies demonstrate the sort of tunability of the drug interactions. In 12-month-old females, 3 hours after treatment, we saw a normalization of serum calcium with both 40 and 80 microgram per kilogram. This suggests that perhaps a calcimimetic allows for the use of higher doses of PTH in aging animals. And so to assess the changes in trabecular bone parameters, we performed high-res CT analysis on distal femurs of the mice. And the data showed that here's, for example, PTH alone in blue, it modestly increased the trabecular bone mass, or the BV over TV there on the left, by 17% and trabecular thickness by about 8%. Although the calcimimetic alone had no effect on these bone parameters, when it was co-injected with PTH 1 to 34, there was a robust increase in the trabecular bone volume, BV over TV, of about 31%, and trabecular thickness of about 18%, which is about double the effect of PTH treatment alone. So in contrast with 12-month-old females, we needed 80 micrograms per kilogram dose in the combined treatment to see an increase in trabecular bone parameters of the L5 vertebrae, indicating age and perhaps sex differences in the drug responses here. And next, we looked at cortical bone of the tibiofibular junction, or the TFJ, which is known to be less sensitive to PTH treatment alone. So indeed here, there was no change between vehicle controls versus PTH treatment alone in the all cortical bone parameters, tissue volume, bone volume, and thickness. And calcimimetic alone actually decreased these parameters, likely due to that kind of chronic hypocalcemic condition when you're on a calcimimetic. When it was co-injected with PTH, it increased in all three cortical parameters, indicating cortical bone expansion. Likewise, in 12-month-old female mice here, we saw significant increases in cortical bone parameters only at the 80 microgram per kilogram dose, again indicating the age and perhaps sex difference in the drug responses. We also performed mechanical testing, which showed increased bone strength in the femurs of 3-month-old male mice injected with both PTH and the calcimimetic versus those injected with PTH alone or vehicle, as indicated by that significant increase in the ultimate force to failure in the co-treated bones. And that just suggests that there's the osteoanabolism induced by the combined treatment produces stronger bone that may resist fracture. So far, we conclude from these studies that co-injections of the calcimimetic can not only sort of abrogate the PTH-induced hypercalcemia, but we also enhance the osteoanabolic effect of PTH by about twofold in both trabecular and cortical bone sites in both young and older adult mice. But the question is, can we do better? Can we use this combined regimen with a stronger designer PTH analog, LA-PTH, LA stands for long-acting, to produce more robust skeletal anabolism, which could possibly build bone in a shorter amount of time to avoid long-term adverse effects and perhaps even cut the costs. So the LA-PTH analog consists of a mutated amino acid sequence of PTH 1 to 34 and PTH-RP. Were first made by scientists at the endocrine unit at Mass General Hospital. And we get this constant supply from JP Villardega, our friends at University of Pittsburgh. And this long-acting PTH analog induces prolonged endosomal CAMP signaling, cyclic AMP signaling, which sustained hypercalcemia for several hours. It also produces a very robust skeletal anabolism when used by itself. It's currently in clinical trials to treat hypoparathyroidism. Its severe hypercalcemia effect, however, prevents its use for treatment of bone diseases. And so we hypothesize that concurrent activation of the calcium sensing receptor with calcimimetic can also mitigate the kind of super hypercalcemic actions of long-acting PTH to allow for more robust skeletal anabolism than a combined PTH 1 to 34 calcimimetic treatment. So indeed here, you can see that like PTH 1 to 34, injections of the same dose of long-acting PTH produces a significant increase in serum calcium, which lasts longer than 12 hours versus like less than 6 to 8 hours with PTH 1 to 34. So data is not shown here, but if we plotted it out, the hypercalcemic effect actually lasts like half a day, 12 hours worth, versus a very short period with PTH 1 to 34. And so co-treatment with the calcimimetic completely normalizes the serum calcium to the level that's very indifferent from the vehicle group, demonstrating that activating calcium sensing receptors effectively counteracts that hypercalcemic actions of long-acting PTH. Furthermore, we also performed behavior testing on mice after 3 weeks of intermittent injections of long-acting PTH and long-acting PTH with a calcimimetic. You can see here that treatment with LA-PTH alone, mice displayed an inability to kind of remember or recognize new objects. However, when combined with the calcimimetic, this decline of cognitive function was rescued. So the mice's short-term memory was definitely affected with intermittent LA-PTH dosing, and it was rescued once you combined it with a calcimimetic. So as we showed earlier, combination of PTH 1 to 34 with the calcimimetic resulted in a higher increase of trabecular bone mass in the distal femur that is about double of that of PTH 1 to 34 alone. In this study, it was about 22.8% versus 10.8% in the single treatment. And as reported previously by the MGH group, treatment with long-acting PTH alone resulted in an over 200% increase in bone mass, which is clearly evident here in this 2D and 3D reconstruction images, where the new bone packed the marrow cavity along with decorticalization of the surrounding cortex. But when you co-inject with calcimimetics, which normalizes serum calcium, it retains the ability of LAPTH to stimulate new bone formation, but has less impact on the surrounding cortex. And so these skeletal responses could essentially allow for further development of regimens for faster bone rehab and perhaps with lower doses and less frequent administrations of the drug versus how we do it now with intermittent PTH, where you have to come in the hospital and get this single shot once daily. Here show von Cossa staining confirmed for micro-CT findings. It shows increased trabecular bone mass, thickness, and number. And to further study this kind of osteogenic expansion, we also measured dynamic histomorphometry with calcine and dimethylcycline double labeling. We found that treatment with PTH and LAPTH alone increased both mineral apposition and bone formation rates, shown here and here. And combined treatment with calcimimetic increased these same parameters, but to a lesser degree, perhaps indicating the tunability of bone formation with our regimen. Next, we measured, furthermore, while trap staining parameters show an increase in erosion surface and osteoclast number per bone surface with PTH and LAPTH treatment alone. When you co-inject with the calcimimetic drastically, this essentially decrease these parameters, indicating an overall inhibition of the resorptive activity of the osteoclast by the calcimimetic. And this also kind of suggests how sort of our combination treatment provides a sort of window for bone formation to outpace bone resorption, thus resulting in more osteoanabolism. At the TFJ, LAPTH with and without calcimimetic increased overall bone volume and bone fraction compared to the control group. And zooming into the 2D and 3D reconstruction or micro-CT images here, you see these kind of bony extensions from the cortical endosteum and periosteum, which highlights the more robust cortical expansion effect of LAPTH compared to PTH by itself, or PTH 1 to 34 by itself. And you can also see the increased cortical porosity, which suggests a surge in perilacunar remodeling by osteocytes with LAPTH treatment. But these changes in cortical porosity was less evident when you co-inject with the calcimimetic. And so with help from Crystal Yee and Tamara Allison at UCSF, we generated data for osteocytes, correlating osteoid volume, osteocyte volume and density to their activity. And you look at PTH and LAPTH alone, promoting sort of a osteocytic activity to contribute to the hypercalcemic effect, right? And so the addition of the calcimimetic decreased the osteolytic activity in the LA plus calcimimetic group. As you can see, it's kind of decreased here. And so we also used nCounter to generate a heat map of a panel of genes known to sort of mediate an osteolytic actions of the osteocyte. We used RNA from marrow flushed with bone here, and the LAPTH column showed this significant increase in those genes like MMP9, 13, and cathepsin K, among others, while the addition of calcimimetic kind of decreased their expression. So certainly the addition of the calcimimetic can have an inhibitory effect on the osteolytic actions of the osteocyte as well, contributing to that decrease of hypercalcemic side effects of PTH or long-acting PTH. And so our second conclusion, we found that co-injections of the calcimimetic normalize the hypercalcemic effects of long-acting PTH and produces an even more robust osteoanabolism than in combination with PTH 1 to 34. In regards to sort of a clinical relevance here, you can imagine the potency of this combined therapy once you can kind of fine tune it to possibly allow for a shortened course that can cost effectively treat patients. And so this is our sort of working model of how the calcium sensing receptors work locally in bone in response to intermittent PTH and long-acting PTH. PTH or long-acting PTH recruits osteoprogenitors and promotes survival of osteoblasts and stimulates periauricular remodeling activity of osteocytes embedded into the bone matrix. Osteoblasts then produce kind of other factors like that rank ligand that recruit stem cells and promote their differentiation at the osteoclasts, which resorb bone and release calcium into the sort of local surrounding environment, which goes into circulation, contributing to the hypercalcemic effect. The increase in this kind of local calcium, particularly under calcium sufficiency, activates calcium sensing receptors to drive that terminal differentiation of the recruited osteoblasts and promote matrix mineralization. Meanwhile, the very same increased calcium sort of has an inhibitory effect in both osteoclast survival and differentiation. And this is where we kind of play with the calcimimetic to help inhibit the osteoclast. And together these favor bone formation. So it's coming to the end of my talk, and I think it's interesting to look back and to see where science has been and where we're going. I've included a few early landmark papers here on the screen, and we're kind of approaching that 100 years of PTH research, and we and others in science are still sort of unraveling this PTH paradox in terms of its mechanism of action. We have this sort of hormone that doesn't always lead to bone resorption, as stated here, but may sometimes produce bone apposition. And so I wanted to sort of thank past and current members of the endocrine unit at the SFBA who have contributed to and supported this project. Wenhan and Dolores for their supervision and support during my PhD and postdoc training, Chaoling for her work with their RNA data, Zicheng and Fu Qing, who contributed to the histomorphometry data, our collaborators and friends from the Villardega lab at UPitt, and other PIs at the SFBA, Anne, Bob, and Dan for their insights, and Alfred and Nick, who run the micro-CT core here at the SFBA. And with that, thanks for your attention, spending a lunch hour here. Ended the talk early, trying to get a lot of thoughts and questions from you guys. So I welcome them now.
Thanks, Christian. That's a very nice talk. And I assume that I will be moderate the Q&A section. So if anybody have a question, either raise your hand under the reaction button, or you can just send through the chat. Christian, can you unshare your slide?
Sorry about that.
No problem. Great.
So this is Matt Warman. Is it possible for me to ask a question?
Oh, yes, go ahead, Matt.
Hi, so it's a great talk, really beautiful data. My question is, how did you choose the novel object recognition test as the neurobehavioral phenotype to look at in the animals getting the long-acting PTH? What led you to that test as opposed to all the other tests that are out there?
Sure, Wenhan, if you can elaborate on that, I'm passing this over to you.
The reason use new object recognition is in our hand, we usually routinely use two assays, Y maze and the new object. And the new object actually is working quite well to pick up more subtle the difference in terms of their cognitive functions. So that's why we picked this. And Y maze a little bit, it's been rougher. So, and I think there are tons of other methods like water maze, those techniques, which is more tedious. And this is just a first pass to look at the effect of the long-acting PTH on the cognitive activities in the mice. Does that answer your question, Matt?
Yeah. You did 5 animals of each. Is it a reproducible finding? Have you done it again? And is hypercalcemia associated with that impairment, or was that just a one-time experiment?
This experiment is reproducible, yes.
That's a really interesting result.
Yeah, but you have to remember this animal is, we put a lot of the LAPTH in there. This is about 40 microgram per kilograms. So, you know, it's really not consider the amount of the LAPTH being used in clinical trial is way beyond that. So that's something to think about. Okay, and Mike, please go ahead.
Yeah, thank you, Christian. Excellent talk. You mentioned the PTH paradox at the end, when we think that constant levels of PTH might increase the bone resorption more than bone formation, and intermittent PTH helps the bone. Here you're using the R0 ligand LAPTH that was developed mainly for increasing calcium, as you have mentioned. How do you, yet you when you decrease the serum calcium, you're able to see a almost stronger bone anabolic response. How do you think about this paradox in your case?
I don't seem to understand the question, the second part where you...
Yeah, so the long-acting PTH seems to imitate more what we call primary hyperparathyroidism, where the PTH level is constantly elevated, which typically is not bone anabolic at the end. Here you have a strong bone anabolic action. How do you explain that?
The mice are... That's a good question. So, I mean, the mice are in constant... It's still intermittent in terms of, it's not like an infusion of this LAPTH. They get kind of a break despite the kind of super physiological injections that they get, right? This isn't constantly being infused into the mice. And so as intermittent as a super physiological dose of a very strong anabolic agent can be, I guess it supposedly still gives the mice time to kind of decide what they want to do with that calcium. You know, like calcium is very tightly regulated physiologically by a bunch of different organs and by the calcium sensing receptor there. And so, and the mice are constant calcium sufficiency. It's not that we're, you know, knocking out the calcium receptor or keeping them under sort of a low calcium diet. And even if that were the case, calcium is so tightly regulated here. And so, yeah, I'm kind of that's kind of my explanation in terms of how this LAPTH is still sort of an anabolic agent in that case.
Yeah, it's interesting.
So Michael, one thing to add is that recently Jean-Pierre Virdec demonstrated that there's a difference between the endosomal and the membrane cyclic AMP signaling pathway. So if you think about the chronic hyperparathyroidism, the ECD receptor signaling both of them, but the drug, the LAPTH is only mainly stay in the endosomal cyclic AMP. So that might produce different skeletal effects.
Yeah, thank you.
Just
Hank?
well,
I just wanted to say thank you. I'm like the others, very impressed by the data. I think it's really pretty amazing. And I think it's an example of our using this intermittent PTH thing is, I think, a bit overdone. We don't know that it's important that it's intermittent, and we don't know why it's important if it is important that it's intermittent. I think it's entirely possible that continuous PTH with the appropriate inhibition of bone resorption might be anabolic. All the anabolic mechanisms don't depend on the absence of PTH for a while. So I think, you know, I think we should be pretty flexible in our thinking about what intermittent is all about, and to be completely prepared for the possibility that it's not needed at all for some of these unusual circumstances, like putting in a calcium sensing receptor or doing something else to block resorption. So for there may be a lot of different ways to use continuous PTH and still build bone. I don't think there's anything in the laws of nature that makes that at all impossible. And I think this is just an example of that.
Yeah, I think in supporting with Hank's ideas that we generated quite a bit of the hyperparathyroidism mouse models. And those models actually is mostly it's anabolic. We see the bone is going up, the densities go up, the bone become bigger. And I think it's because the animals are housed in eating the chow, which actually have more than enough calcium available. Jean-Pierre, please.
Yes, I will just comment on your question, Michael. So the PTH receptor and the calcium sensing receptor, they interact functionally. So the calcium mimetic, in fact, is redirecting the signaling mediated by LAPTH. It turned the LAPTH into a biased ligand that favor bone formation instead of the calcimic response. The molecular level, for example, the calcium mimetic will block the recruitment of arrestin to the receptor, so it biases the intracellular signaling mediated by PTH receptor.
Yeah, it's very impressive data, Christian. Thank you.
Harold, you have a question? Yeah.
Yeah, this is Harold. Very nice data. So I unfortunately missed the first part of your talk, but did you have a chance to compare the effects of a bisphosphonate versus the calcium mimetic drug? Do you get the same anabolic response if you now give LAPTH on top of that?
We haven't tested that yet. I'm not sure if we have plans to actually even test an anti-resorptive in combination with LAPTH. It'd be nice to show, sort of what you can do with like a short burst of LAPTH and then kind of transfer them to a bisphosphonate if you can, and try to mitigate sort of the immense amount of bone that LAPTH can actually build. So for example, when we inject these mice after 4 weeks, they're sort of unhealthy and with LAPTH by itself. Like again, they do super physiological sort of doses with these animals. You can see that kind of the marrow space is very much closed when we're showing the bone data, and so the mice are kind of sick. So if there's just kind of a way we can kind of fix the regimen such that you kind of go back and forth to maybe perhaps retain that bone. That would be nice to see, yeah. But unfortunately, we haven't tested that in any mice.
I mean, what Jean-Pierre was saying, that there's synergism between the calcium sensing receptor and the PTH receptor when activated, suggests that bisphosphonate would not have the same impact that you see with a calcium mimetic.
Yeah, exactly. I think I agree. I think because the target organs is very different. Bisphosphonate most work on the bone, but the calcium receptor, PTH1 receptor interaction, it happened in multiple organs, for the calcium absorption, excretion, and then mobilization of the calcium in the bone. It's integrated. So I would predict the different result if you use, the different drug, calcium mimetic versus the bisphosphonate. Hank, please.
Yeah, I think this is a very interesting and lively discussion. I think it's fair to say that Jean-Pierre's observations may well be an important part of the story, but he doesn't know that. Nobody knows that, because what you need to do is do controlled experiments to see how much blocking osteoclast versus effects on the various multiple different mechanisms that PTH uses to increase bone mass. And so I think that it may be because it's a biased agonist. That may well be part of it, maybe the whole story. But I think the next part of this story is to find out why this combination works. And I think it's important to understand that that's an unanswered question right now.
Right, and we do have plans to sort of to knock out the calcium sensing receptor in the osteoblasts, and if possible, the osteoclast using a cathepsin K knockout, if that's possible.
Yeah, good.
Deborah?
Great talk, Christian. And there's also a question in the chat, which maybe I'll just read it and then ask my own question as well, from Zaina Salut. Thank you. I was wondering whether it has been studied if the calcimimetics combination with PTH decreased the oncogenic effect of PTH alone.
We haven't, there are no studies on that. We only treat our mice for 4 weeks. The mice that they were treating back in those early studies of intermittent PTH was way longer than that. So the answer is kind of, we don't know. Yeah.
And then the question I had for you was about your transcriptomic analysis. And I was wondering if you saw any signatures of genes or pathways that were not rescued by the calcimimetic in the LA PTH group.
So we ran the N counter on marrow, on bones that had marrow in it as well. In terms of like, we have like an osteo panel that we see. I didn't show the data here, but you can see, we essentially see kind of the trend of a signature trend towards kind of mineralization of the bone. The kind of, so when we treat with LAPTH, kind of like the genes just kind of explode. All of the osteo positive type of genes kind of just are all on. But when you treat it with calcium sensing receptor, it kind of it focuses it towards kind of the kind of bone maturation, mineralization type of genes that turn on. I wish I included it into here because it's quite beautiful when it kind of blows up and kind of fine tunes it. And you can only see a certain amount of genes that are very important, that are significantly kind of chosen to be upregulated. We've done it with both like kind of acute treatment. We did it with sort of a 3-hour treatment to kind of see just the initial earlier genes. And then we also did it after 4 weeks of treatment where the bone has really been changed. You see it, you see kind of the more focused, like I said, more focused genes. Well, I'll show you that picture. I'll email it to you so you can see.
Thanks, Christian.
So I think we about have all the time we have, and we need to pass on to the next section. Will be introduced by Dr. Yingzi Yang, I assume.
Yes, that's right. We'll go on now to the next part of the session. This was really quite a spectacular talk. One of the things that's missing with the Zooms is a thundering applause kind of effect that we can't have when we have when we're not in an auditorium. But that was a spectacular talk and a wonderful discussion. So thanks to everybody for that. And just as you said, Yingzi Yang will introduce the next speaker.
Hi, everyone. It's my pleasure to introduce Qian Cong, a postdoc fellow in my laboratory, to talk in the second session. Qian joined my laboratory in the summer of 2017. Since then, she has been working on the project identifying cellular and molecular mechanisms underlying heterotopic ossification, both genetic forms and injury-induced non-genetic forms of heterotopic ossification. And she has done a fantastic job finding a novel self-amplifying, self-propagating loop between yap and Sony hedgehog, and that is fundamentally important in driving the formation and extension of heterotopic ossification, which is pathologic bone formation in soft tissues. More recently, she is working on identifying the cell lineage in regulating or in contributing to the ectopic bone formation in soft tissue. Without further ado, Qian.
Yeah, I will share my screen. Okay. Okay, can you see my screen?
Yeah.
Yeah, thanks for this introduction, and thanks for the CSR organizer for giving me the opportunity to present my work today. Mechanistic and the therapeutic studies of initiation and expansion of heterotopic ossification. So I will introduce a human disease called heterotopic ossification. It is an ectopic bone formation in extraskeletal tissue. There are two types of HO, genetic HO and acquired HO. The major two forms of genetic HO are POH and FOP. Most cases of POH are caused by inherited inactivating mutations of genes. It occurs predominantly by intramembranous ossification, which osteoblast differentiation is directly from mesenchymal progenitor cells. In the human patient, initially bone formation occurs under the skin, and then it extends progressively into deep connective tissues and the skeletal muscle. Our previous work showed that in the POH mouse model, gene deletion in adult mice induced by subcutaneous adenovirus pre-injection resulted in subcutaneous ossification, and the HO invaded and expanded to the deep muscle, which is similar to that found in the POH patient. And this is the mouse model used in our work. Another genetic HO is FOP. FOP is caused by activating mutation of BMP type 1 receptor, ACVR1. It occurs by endochondral ossification, in which mesenchymal progenitor cells first form a chondrocyte, then they form osteoblast, which is different from intramembranous ossification in POH. The two different ossification forms govern all bone formations. The classic forms of FOP are heterozygous mutation of R206H. It is the classic mutation found in 97% of human FOP patients. Another transgenic mouse line is Q207D. It allows inducible expression of an artificial constitutively active variant of ACVR1 with robust HO production. We utilized these two FOP mouse models in our work. The genetic HO contains intramembranous and endochondral ossification. So understanding the important cellular and molecular mechanism provides critical insights into more common non-genetic conditions. Acquired HO appears to be regulated by both endochondral and intramembranous bone formation and occurs frequently as a complication of trauma, fractures, total hip arthroplasty, deep burns, and central nerve system injury. This is a case that HO formed around the right hip. It is caused by hip arthroplasty, and the HO is progressively increased from 5 months to 10 months post-surgery. At present, the clinical therapy is limited to non-steroidal anti-inflammatory drugs, local irradiation, and surgical excision with a high recurrence rate and complications. Thus, HO is a serious and costly complication and the major unmet medical challenge. There are two kinds of trauma-induced HO mouse models. One is Achilles tendon puncture, ATP mouse model, which is utilized in our work. We used 4 weeks old wild-type mice. Achilles tendon was punctured with a needle from lateral aspect, and the puncture was repeated multiple times at different parts of the Achilles tendon. In this repetitive healing after injury, ectopic bone formed. Another injury-induced HO mouse model is pterotomy with trauma. So first, the mice were burned on the back, and then the tendon was injured. The HO will be detected around the tendon, which is severe than the ATP mouse model. So since the current treatment for HO relies heavily on surgical removal of ectopic bony tissues, which causes additional trauma and thereby easily leads to recurrent HO. So in the face of such intractable clinical problems, the first question we asked is, what is the cellular and the molecular mechanism underlying the genetic and the non-genetic HO? So in order to understand the mechanism of this disease, first we set up POH by using GNS flux mice with one-shot adenovirus-free local injection in the subcutaneous region. In 6 weeks, 3 months, and 8 months post-injection, we performed the micro-CT. The HO is progressively increased from 6 weeks to 8 months. We sectioned the samples after micro-CT, and the wound bone was detected by the von Causa staining. After Cray injection 6 weeks, the ectopic bone formed in the subcutaneous region where the virus was locally injected. And after 3 months and 8 months, it invaded and expanded to the skeletal muscle. So that is exactly what has been found in the human POH patients. And consistently, osteoblast markers, CAL1 and ostrix were increased. There are 2 possibilities for this phenotype. One is HO expansion is caused by expansion of GNS mutant cells proliferation, which is a cell autonomous event. Another is GNS mutant cells recruit wild-type cells to the progressively forming bone, which is a non-cell autonomous event. So to distinguish between these 2 possibilities, the GNS mutant cells were labeled by the TD tomato, and osteoblast cells were labeled by the GLP. Interestingly, many GLP positive cells are not TD tomato positive cells, where the number of TD tomato positive cells was similar in the control and the knockout groups. So therefore, more TD tomato negative cells were induced by the GNS mutant cells to become osteoblast, contributing to HO expansion, confirming that the ectopic bone expansion is a non-cell autonomous process. So to further confirm this non-cell autonomous process induced by GNS mutant cells, we performed the conditional medium experiment. So firstly, we isolated GNS flux subcutaneous progenitor cells called SMP cells. They are potential osteoprogenitor cells within the subcutaneous fat connective tissue, which could be induced to become osteogenic cells and depocytes. We infected the cells with GLP or Cray virus and collected the conditioned medium every 2 days and added the conditioned medium to wild-type SMP cells, followed by ALP and von Causa staining. The GNS mutant conditioned medium promotes the osteoblast formation and the ossification, and the expression of osteoblast markers were increased. So what is the secret factor in this conditional medium? So previously, we showed that activated heat-shock signaling was both necessary and sufficient to induce HO in POH, and therefore reasoned that soft tissue cells could help be induced to become osteoblast via activated heat-shock signaling. And to test this hypothesis, we marrowed the candidate heat-shock ligands. Sunny heat-shock and the Indian heat-shock are upregulated, especially sunny heat-shock, while desert heat-shock doesn't change. And the sunny heat-shock protein level was also highly increased in the GNS mutant conditional medium. To determine whether increased sunny heat-shock plays an essential role, we generated sunny heat-shock flux, GNS flux SMP cells, and both sunny heat-shock and GFS were removed by adenovirus Cray infection in vitro. And sunny heat-shock loss reduced osteoblast differentiation. Furthermore, removing 1 copy of sunny heat-shock in vivo drastically reduced the GNS mutant induced HO, whereas removing both Sony Hedgehog copies further reduced HO with almost a complete rescue. The next question we ask is the mechanism driving Sony Hedgehog ectopic expression. The gene expression in wild type and the genus mutant cells were analyzed by RNA-seq. The hippo signaling was upregulated in the genus mutant group, and osteoblast differentiation, ossification, and bone mineralization were increased in geoanalysis. And we are excited to say that the expression of YAP target genes, CTGF and CYR6Y1 were increased in the genus mutant cells based on the RNA-seq data. And to confirm the role of YAP in ectopic bone formation, we removed the YAP and the genus by adenovirus Cray infection in vitro, like removing Sony Hedgehog. Loss of both YAP and the genus reduced osteoblast differentiation and Sony Hedgehog and Indian Hedgehog expression. And furthermore, we removed the YAP in vivo. Partial loss of YAP drastically reduced the HO caused by genus loss. So while complete YAP loss more effectively inhibited HO at 6 weeks and 3 months after adenovirus Cray injection. Since YAP loss halted HO, we tested whether HO can be inhibited by the YAP inhibitor VP, which interferes YAP-TID binding that is required for YAP-mediated gene expression. YAP was applied topically or while IP injection to the genus Flox mice immediately after adenovirus Cray injection. Both delivery method inhibited HO, with IP injection being more effective. So as we have found that both YAP and the Sony Hedgehog were much increased in gene S mutant cells, and they are required for HO formation. In addition, the expansion of ectopic bone is induced by a non-cell autonomous process. So we need to confirm that both of YAP and Sony Hedgehog are involved in this non-cell autonomous process in vivo, regulating ectopic bone expansion, and then to test the relationship between YAP and Sony Hedgehog. So firstly, we performed YAP and Sony Hedgehog immunostaining. YAP and Sony Hedgehog expression were upregulated in genus mutant cells labeled by the TD tomato after Cray injection. And interestingly, many YAP positive cells and Sony Hedgehog positive cells are not TD tomato positive cells. These cells are not genus mutant cells. They are surrounding wild type cells. So this data suggests that both YAP and Sony Hedgehog are activated non-cell autonomously to drive expansion of osteoblast differentiation during HO progression. Then we performed the conditional medium experiment to determine whether YAP could regulate Sony Hedgehog expression and secretion. The conditional medium from genus mutant, genus YAP double removal, and the genus Sony Hedgehog double removal SMP cells was applied to the wild type SMP cells, while the genus mutant medium promoted osteoblast differentiation. Such activities were similarly abolished in the conditional medium from YAP loose cells and the Sony Hedgehog loose cells. And as expected, YAP deletion in SMP cells reduced Sony Hedgehog protein levels in the conditional medium. So this data indicate YAP activation is required for Sony Hedgehog expression and resulted in Sony Hedgehog protein secretion to recruit other cells to become osteoblast. To determine whether YAP and the Sony Hedgehog can regulate each other, we perform immunostaining. In the Sony Hedgehog removal mice, the ectopic YAP expression was much reduced compared to this panel. And similarly, in the YAP removal group, ectopic Sony Hedgehog expression was abolished, demonstrating that YAP and the Sony Hedgehog regulate each other in vivo, therefore triggered a positive feedback loop resulted in HO expansion. So YAP is required to active Sony Hedgehog expression downstream of GNS. The next question is whether YAP is a direct transcriptional factor of Sony Hedgehog. We are very interested in understanding the chromatin remodeling occurs during cell fate changes. So we performed ATAC-seq to determine chromatin accessibilities in wild type and the GNS mutant SMP cells. We found that the identified peaks corresponding to Sony Hedgehog promoter and enhancer. And the signal intensity was strongly enhanced at the Sony Hedgehog promoter and the enhancer in the GNS mutant group. And to test whether Sony Hedgehog is a direct transcriptional target of YAP, we performed a cheap experiment to investigate the interaction between YAP-T's protein and the Sony Hedgehog DNA. So several YAP-TIS4 binding sites were identified and confirmed by the qPCR analysis in these regions indicated with the red boxes. So this data demonstrate that Sony Hedgehog is a direct transcriptional target of YAP. To determine whether the identified YAP-TIS4 binding sites are required for Sony Hedgehog expression, we utilized the CRISPR-I technique to block the binding of TIS4 to Sony Hedgehog promoter and enhancer, therefore inhibiting the Sony Hedgehog gene expression. We set up a contrast contains U6 promoter, guidon A, and endonuclear dead Cas9 crab fusion protein, and packaged it to the lentivirus. So after infection, the dead Cas9 crab protein will occupy the region guiding by the sgRNAs. For our experiment, it will occupy the specific TIS4 binding sites, which were identified by the qPCR. The two binding sites on Sony Hedgehog promoter and the two sites on Sony Hedgehog enhancer, therefore blocking the binding of TIS4 protein to Sony Hedgehog promoter and enhancer. As expected, the TIS4 binding sites blocking significantly reduced the Sony Hedgehog expression in the genus mutant SMP cells. So taken together, this data demonstrate that Sony Hedgehog is a direct transcriptional target of YAP. Another major form of genetic HO is FOP. We have known the mechanism driving ectopic bone expansion in POH mouse model. So however, we don't know whether the identified mechanism can be applied to FOP mouse model. So we set up FOP mouse model by using ACVR1, Flox, Scalopsis cri, which is tendon-specific cri mice. So there is ectopic bone in the tendon area. And the hedgehog signaling YAP target genes and the Sony Hedgehog, Indian hedgehog are upregulated. Consistently, YAP and the Sony Hedgehog were increased in the tendon based on the immunostaining. Interestingly, we also detected many YAP positive, OPN negative, and the Sony Hedgehog positive Ostrix negative cells, suggesting these cells were primed for differentiation into osteoblast. Moreover, we also set up another FOP mouse model by using ACVR1 R206H Scalopsis cri mice, which is a more common mutation in human patients. The ectopic bone was detected in the tendon of the 3-month-old mutant mice, and the YAP target genes hedgehog signaling and sonic hedgehog were much increased in the 3 months old mutant group. And interestingly, the increased YAP target genes, sonic hedgehog, and hedgehog target genes were detected even in the 1.5 months old mice before ectopic bone formation. And we next tested whether HO can be inhibited by the YAP inhibitor VP. The experiment was performed by Dr. Paul Yu's lab. The VP was applied via IP injection to the ACVR1R26H skeletal screen mice for 8 weeks. The HO was inhibited with VP injection. And then we generated sonic hedgehog flocks ACVR1 Q207D and YAP flocks ACVR1 Q207D mice with adenomycin screen and carbapenem factor intramuscular injection. Sonic hedgehog and YAP deletion abolished the ectopic bone formation, and the bone markers, hedgehog target genes were downregulated. So HO contains genetic HO and injury-induced HO. The genetic models provide critical insights into more common non-genetic conditions. We asked whether YAP and sonic hedgehog are also required for injury-induced HO. So for this, we set up a trauma-induced ATP mouse model. So the heterotopic bone formed 6 weeks after puncture and continued to enlarge for up to 12 weeks. And the YAP target genes, sonic hedgehog, Indian hedgehog target genes were upregulated. And consistently, Achilles tendon showed strong YAP and sonic hedgehog expression after surgery. And to confirm the essential role of YAP and sonic hedgehog in HO, we set and sonic hedgehog flocks mice and removed the YAP and sonic hedgehog by injecting adenomycin screen to the Achilles tendon. The micro CT results showed less HO after injury in the YAP and the sonic hedgehog removal groups, suggesting that YAP and the sonic hedgehog are required in ATP mouse model. So since YAP or sonic hedgehog lose altered HO, we tested whether HO can be inhibited by the YAP inhibitor VP and a sonic hedgehog monoclonal antibody in the ATP mouse model. And the VP and a sonic hedgehog monoclonal antibody was applied via IP injection immediately after ATP surgery for 6 weeks. So both of them inhibited HO. So this is the summary. A post-adenomycin screen injection or the ATP injury, YAP and sonic hedgehog were upregulated. And YAP activated cells secrete sonic hedgehog to the environment, leading to YAP activation and sonic hedgehog expression in the surrounding cells. And the surrounding wild type cells will secrete sonic hedgehog, further induce YAP activation, sonic hedgehog expression, and osteoblast differentiation in more stem and progenitor cells. So this forms a self-amplifying and self-propagating loop leading to ectopic bone growing bigger and bigger. It's more important, this YAP sonic hedgehog self-amplifying loop in ectopic bone formation and growth was first found in the POH mouse model. And it was also found to operate in several FOP mouse models. As POH and FOP are two genetic disease wherein HO arises by distinct ossification mechanisms. So it is notable that we also demonstrated that both YAP and sonic hedgehog are essential for injury-induced HO, which includes a mixture ossification mechanism of both intramemory and endochondral ossification. Thus, we found a shared common cellular and molecular mechanism underlying all forms of HO. However, there's still poor understanding of the cell origin of ectopic bone, which limits the treatment or prevention of ectopic bone formation and expansion. So our next question is, which stem cell population contribute to ectopic bone formation in POH, and whether the mature muscle cells could also be reprogrammed to form ectopic bone in POH. So previously, Dr. Leva's group identified the stem cells contributing to ectopic bone in Heterotopic mouse model, which I have introduced in the previous slides. This is one of the injury-induced HO models. And they isolated the tendon cells at different time points and performed the cell RNA-seq. They found that PDGF receptor alpha positive and PX1 positive cells are the major stem cell in tendon. They also showed the staining of PDGF receptor alpha positive cells, that is the major stem cell population contributing to ectopic bone formation. So in our POH mouse models, we found that the ectopic bone invaded into the deep muscle through muscle interstices. So we stained the PDGF receptor alpha antibody, and at the same time using the PDGF receptor alpha CRE-ER TD tomato mice with tamoxifen injection for 6 weeks. Both of the methods showed that PDGF receptor alpha positive cells locate in the subcutaneous region and the muscle interstices, so where the ectopic bone formed and invaded. And to test whether PDGF receptor alpha positive MSA contribute to ectopic bone formation, we utilized the PDGF receptor alpha GLP genus flux mice with adenovirus CRE injection. So the upper panel is the control, the lower panel is 6 weeks post-injection. We found ectopic bone formation in the subcutaneous region by the micro-CT. And the PDGF receptor alpha positive cells co-localized with the TD tomato. Moreover, the PDGF receptor alpha positive cells are increased in the genus mutant group. The 3 months post-CRE injection, we found much more PDGF receptor alpha positive cells contribute to ectopic bone formation in the deep muscle. And to further confirm that PDGF receptor alpha positive cells are the major source for ectopic bone, we utilized an inducible lineage tracing mice, GFS flux, PDGF receptor alpha, CRE-ER, TD tomato with tamoxifen injection for 6 weeks. We found that PDGF receptor alpha lineage cells were induced to become osteoblast in the tibia. And to our surprise, we also found ectopic bone in the ventral subcutaneous region, shown in this schematic here. And here's the micro-CT showed that ectopic bone. And I also did the Vencosa staining to confirm that this is bone. And we extracted RNA and performed the qPCR from both tibia and the ventral subcutaneous region, found increased osteoblast markers and the YAP target genes. And the immunostaining of ostrix showed the colocalized of PDGF receptor alpha positive cells in the subcutaneous region and the tibia, in the tibia. Yeah, this is the tibia subcutaneous region and the ventral subcutaneous region. So this state, so the green is ostrix positive cells. So this state showed PDGF receptor alpha lineage cells were induced to become osteoblast in HO, suggesting that PDGF receptor alpha positive MSA are involved in HO formation. And in order to test the essential role of PDGF receptor alpha in vivo and in vitro, we injected PDGF receptor alpha inhibitor, imatinib, for 6 weeks in the POH mouse model and found much decreased HO. And the in vitro study imatinib treatment inhibited osteoblast differentiation promoted by the GNAS mutant. So, however, compared to the POH mouse model with adenomyos cray injection, in which cray was expressed subcutaneously with no lineage specificity, so loss of GNAS in the PDGF receptor alpha positive cells resulted in smaller ectopic bone, which raises the possibility that other cell populations may also contribute to HO. Since we found that the ectopic bone invaded into deep muscle region 3 months post adenomyos cray injection in the GNAS mutant mice, we tested our hypothesis that the myoblast cells could be reprogrammed to form ectopic bone. So the endogenous flux myld cray ER T tomato mice, tamoxifen injection induced GNAS loss in the myoblast lineage. We collected the mice 6 months and 9 months after tamoxifen injection. However, we didn't find ectopic bone. So, confirmed by our immunostaining of the ostrix and the markers of the osteoblast. There's no ectopic bone formation. Then we utilized MCK cray GNAS blocks mice to confirm our findings. MCK is the marker of myotubes, which is later than myoblast cells. So in this 1-month-old mice, we didn't find ectopic bone formation, but I'm still collecting 6 months and 9 month samples to confirm it. Okay, this is the summary. In our study, we found a self-amplifying and self-propagating loop leading to ectopic bone growing bigger and bigger. So during the ectopic bone formation and the expansion, so the PDGF receptor alpha MSA can serve as the ectopic bone progenitors in the POH. And the committed myoblast cells could not be effectively reprogrammed to osteoblast cells in POH. So now we are still doing more experiments to test whether the muscle stem cells, the satellite cells, could be induced to ectopic bone in POH. So at last, I would like to express my deep and sincere gratitude to my mentor, Yingzi, for giving me invaluable throughout my postdoctoral research. Her encouragement and support helped me to accomplish this project and understand how to present the research work clearly. I also would like to thank our collaborator, Dr. Po Yu and Dr. Yu Ji, helping us to set up FOP mouse models and treated them with VP, the YAP inhibitor. And my sincere thank also goes to all the research members of Young Lab. Yeah, thank you. Also, thank you for the CSR organizer. And at last, welcome to join our lab. We are still looking for more postdocs. Yeah, thank you.
Thank you, Qian. That's a great talk. Questions?
Hi, Qian. This is Ed Xiao from UCSF. Thanks very much for a really nice talk. It's good to see you and Yingzi too. Could you clarify a little bit of what are the, I think you called them SMP cells?
Yeah, SMP cells.
What exactly those are and where they're located?
That is the subcutaneous progenitor cells that has the ability to be induced to the osteogenic cells and the adipocytes. Yeah, that's a kind of progenitor cells because of HO formed in the subcutaneous region first after adenomyos cray injection. So we isolated the SMP cells, the subcutaneous progenitor cells, for our in vitro study. Yeah, thank you.
Hank?
This was really, really beautiful work. Congratulations. I had one minor question, really. While there's lots of data suggesting that GS-alpha signaling can suppress hedgehog signaling in a variety of ways, I just don't know what the story is about GS and YAP. Is it you're thinking that hedgehog is the main target of the GS-alpha knockout, or is there an effect on YAP as well?
Yeah, I think from our previous study that we found that GFS lose activated hedgehog signaling by the PKA. Yeah, because GFS is the activator of PKA, and the PKA is the inhibitor of hedgehog signaling. So GFS can directly, regulate hedgehog signaling by PKA. And in our study that we found that GAP is also activated after GFS lose. Yeah, from the previous leaders, that has several possibilities that there is a paper said that GFS can directly regulate YAP activities in basal carcinoma cells, I think. And another study indicated that GFS can regulate both CLE1 and the YAP transcriptional activities. Yeah, in the basal carcinoma cells. Yeah, I remember that.
Thank you.
Thank you.
Okay. Andrew?
Hi, I really enjoyed this. It was super interesting. I have a couple of questions, and it's pretty related to what Hank was just asking. I guess it is, so I guess I'm looking kind of for a model. Is the model at PKA phosphorylation of YAP is directly keeping it out of the nucleus or inducing its degradation? And the other question is, in a lot of systems where people have been looking at effects of YAP on TEAD, there's YAP and its cousin TAS. And in many instances, you had to do a loss of function for both YAP and TAS to elicit a phenotype. And so my question is, are you surprised that loss of YAP alone was able to reverse the phenotype? Do you think that TAS plays any role in this? Have you looked for any possibility of redundancy in this pathway? Or do you think that they're actually, these two TEAD binding proteins have different activities on the hedgehog, sonic hedgehog expression?
Yeah, that's a very good question. Yes, actually, we only deleted YAP in our mouse model, and we used the inhibitor of YAP. Actually, that is the YAP-TIS interference inhibitor to inhibit the YAP-TIS binding, therefore inhibited the downstream mediated gene expression. So I think that may explain a little bit about that. But if I would like to clarify that, yes, it is necessary to delete TAS along, YAP along, and YAP TAS together to demonstrate that. Yeah, I think that's a good point.
So I just want to add, so YAP TAS appears to be quite they have redundant roles, and they seem to be dose dependent. So it's like a whole pool of YAP TAS in it. I don't think TEAD necessarily distinguish YAP or TEAD, that YAP or TAS as partner. So I guess because this is a self-amplifying system, if you can bring YAP or TAS activator to below certain threshold, it's sufficient to stop it.
Okay, so you think it's a threshold issue. The other alternative is that they actually are not redundant.
Yes, but before we do the experiment, we actually don't know. Yeah.
Got it.
Yeah.
Thank you very much. Wonderful work, just spectacular.
Thank you.
Jay?
Yeah, I mean, great talk. I really love it. I mean, it's so amazing. So much work is there. But I have one question and one comment. Have you ever used the type 2 Cre? I mean, Edward Shao and other groups also show that it's endothelial markers also important for kind of HO precursor cells too. Maybe that's another one is you may try. And then another one is that usually it's FOP and kind of HO, especially trauma-induced HO. Inflammation is one of the major key factors too. And have you ever seen the inflammation is associated with YAP-TAZ pathway?
Yeah, that's a good question. So the first one is tied to, yeah, I think it's a good suggestion. Actually, I tried before, but only tried immunostaining of tied to PX1 in the subcutaneous region after adenovirus Cre injection. And I also tried nesting and SCAR1. Yeah, I think from our data, PDGF receptor alpha is the most important one contribute to HO formation. But I will try. I think that's I will do more experiment to clarify it. Yeah. And another one is the inflammation. Yes, after injury, especially in our injury-induced mouse model, we did say a lot of inflammatory response. Yeah, this is what we are still doing now. Yeah, and I hope to update the progress of our project later, yeah, soon in the future.
Harold?
Yeah, this is really amazing piece of work. And I'm sure you've also questioned how you can reconcile these data in mice with the findings in humans. I mean, I assume that the heterozygous mice, whether they have the GS-alpha defect on the maternal or the paternal allele, don't show a phenotype. But in humans, you obviously have to have a paternal mutation in order to get the heterotopic ossification and POH. So mice and humans are basically very different, and that's the end of the story? Or is there potentially a second mutation that you're inducing through whatever mechanism you're using?
Yeah, exactly. In the human patients, that the heterozygous mutation of genus cause HO. It is much severe than in our mouse model, because in our mouse model, the homozygous mutation causes. I think that might be a little difference between the mice and the human. Mm-hmm. Yeah. Yeah, but in our mouse model, the heterozygous mutation of genus, we didn't see any phenotype. So we always use the heterozygous ones as the control.
It just takes longer. So if we wait for,
it may take longer.
Wait for like 10 months or 12 months, and then we start to see subcutaneous ossification.
So I guess in the heterozygous mice, you see.
Correct. Yes.
And do you see a difference whether the mutation is on the maternal or the paternal allele?
That I don't know. I think somebody published that actually.
Well,
Jimmy, I think at Johns Hopkins. I don't know whether she's still there.
Yeah, she's in Connecticut now.
Okay.
But I think it was.
I can't remember whether there's a male-female difference, but I think she used heterozygous mice, and she waited long enough. She can see subcutaneous ossification.
I mean, that's certainly true, but I don't think there was a major difference between having a maternal or the paternal deletion.
Yeah, but in order to mimic sort of early or pediatric HO like human, in mice we have to delete both.
Yeah.
So can this is Matt, can I ask, in mice that are heterozygous, did you measure YAP and SH levels to see if they're trending higher as well, even though the phenotype is much less penetrant?
Excellent question.
I think it's a good question. We probably to do it. Because, we sometimes we just, to be honest, and I think a lot of times the Qian, because of the breeding scheme, she compared the heterozygous, which has no phenotype, and the homozygote, which has phenotype. You know, I guess we did not, a lot of time we did not include a true wild type. So because the key is, you know, did you see a dramatic difference in terms of gene expression, this and that?
Yeah.
I think we've sort of come to the end of our time, and this has been a great presentation and discussion following a previous great presentation and discussion. So I want to congratulate the speakers and also congratulate the audience for a wonderful discussion and for staying around. So I hope everybody has a great weekend, and we'll adjourn now. Thanks so much.
Yeah, thank you. Have a nice weekend.
Bye-bye, everybody.
Bye.
Great talks, everybody. Thank you.